Hello everybody and welcome to this module on modification interference assays. So I'll be discussing modification interference assays. So the first thing that I want to say is that I've discussed DNase-1 footprinting, and DNase-1 footprinting allows us to identify a region that contains a control sequence, but it does not allow us to identify the precise location of the control sequence. The modification interference assay, on the other hand, allows us to do so. So the first thing that we do is similarly to the DNase-1 footprinting, similar to that or akin to that, we start with end-labeled restriction fragments. So I'm going to draw end-labeled restriction fragments here. So we'll have end-labeled restriction fragments, and so we have them end-labeled here so that we can detect them via gel electrophoresis. And then we perform treatment here. So we perform treatment with a substance like dimethyl sulfate. And what dimethyl sulfate does is that it methylates, so dimethyl sulfate. And what disulfate does is it attaches methyl groups to guanine nucleotides. So what it will do is it will attach methyl groups to guanine nucleotides. So if I redraw the restriction fragments, the end-labeled restriction fragments, let me redraw them down here. So if I redraw the end-labeled restriction fragments, they are end-labeled, as you can see here, they are end-labeled. And what will happen is that you'll have modified guanines, that is methylated guanines, or methylated guanine nucleotides. And so let's say we obtain a methylation right here, and then we obtain a methylation, say, right here. And then we obtain a methylation, say, right here. So these are methylated. These are methylated. So I'll do this. And these are methylated or modified guanines. So modified, and by modified, I mean specifically they are methylated guanines. And then what we can do next is, so what happens here is we have methylation. So we had a methylation right here. We had a methylation on a guanine nucleotide right here, and a methylation on a guanine nucleotide right here, and so forth and so on. And then what we do next is we add the nuclear extract, which contains the regulatory protein. Before I move into that, what I should say here is that this treatment with dimethyl sulfate is done under limited conditions. So this is done under limited conditions. And the reason for that, the reason this is done under limited conditions, is so that one nucleotide is modified per DNA strand. So one nucleotide modified per DNA strand. And then the next thing that you ought to recognize is that we add the nuclear extract. So we add the nuclear extract. We add the nuclear extract. The nuclear extract contains the regulatory protein. It contains the regulatory protein. And the protein is blocked from being bound to the methylated sites. So this is the reason we perform methylation. And so what's going to happen is that you're going to have, I'm going to draw the restriction fragments again. Draw the restriction fragments again. So with the labeled end. And then we have a methylation here, and say a methylation here, and then the same methylation that we saw up top here. And then what's going to happen is we're going to have the bound protein. So we can draw the bound protein. So let's draw the bound protein, say, in white. So we can draw the bound protein. And so you might have a bound protein that binds, say, in this region. This is where the control sequence is. So we'll draw the bound protein in this region. Or I should say, excuse me, let me redraw this for accuracy. Let's say the bound protein binds to this region. So it doesn't bind here because we have a methylated region here, but it's able to bind here. And so let's say this is the bound protein, but here we have an unbound protein because we have blockage by the methylated guanine nucleotide. And then what we can do next is we can perform an agarose gel electrophoresis. So the next step is to perform an agarose gel electrophoresis. So we can perform an agarose gel electrophoresis. And the reason we do that is so that we can separate the bands that contain the bound protein from the band that does not contain the bound protein. And so if we perform an agarose gel electrophoresis, what we will get is something along those lines. Let's say we have the control lane, or I should say the size markers right here. So let's say we have the size markers right here. So let's say we have the size markers right here. And then for the actual bands of interest, whereby we have the test that we perform. So this is the test that we perform. We'll have two bands. So we'll have one band, say, right here. We'll have band, say, right here. And then we'll have another band. I'll draw it in a different color, say, right here. Now this band right here, it's the non-retarded band. So this is the non-retarded band. That is, it has no bound protein. It contains no bound protein. And this is why it was able to migrate at a faster migration rate and move further down in the agarose gel. And then what we can do is we can purify this DNA. So we can purify this DNA right here. And so the next step is to purify the DNA. So we can purify the DNA. We can purify the DNA. And then what we obtain is, and so just to be clear, this band right here comprises of the DNA protein complex. So this comprises of the DNA protein complex. So this is why it had a slower migration rate in the agarose gel, whereas this one contained only the DNA, Now, once we purify the DNA, we'll have something akin to this. So we'll have the restriction fragment. It's end labeled, and it contains the methylated guanine nucleotide, say right here is where we drew it approximately. And then we perform treatment with piperidine. And so what piperidine does is, so next step is we perform treatment with piperidine. And what piperidine does is it cleaves at methylated regions. So it cleaves DNA at methylated regions. And so what will happen here is we will have cleavage of DNA at methylated regions. And then once we do that, once we cleave the DNA at methylated regions with piperidine, the next step that we can perform is a polyacrylamide gel electrophoresis to determine the sizes of this cleaved DNA, that is the DNA that has been cleaved at methylated regions that we purified from the agarose gel. And so what we have here is, say, something that looks like this. Let me draw this in a smaller portion. So if we were to redraw this, this is the DNA cleaved at methylated regions. So it's been cleaved right here. So this is where piperidine cleaves. So this is piperidine's cleavage site. And then what we do is we determine the sizes via polyacrylamide gel electrophoresis. So we perform polyacrylamide gel electrophoresis. And the reason we perform polyacrylamide gel electrophoresis here is so that we determine the sizes of the bands, which help us indicate or deduce the position of the DNA in the DNA fragment of guanines, whereby the methylation prevented the protein binding. And so the guanine that you saw here, this guanine is located within the control sequence. So this is located within the control sequence. And we know that because the regulatory protein was meant to bind there, but it wasn't able to bind there because it was blocked by the methylated guanine that is located there. And so this is what's ought to be recognized here. Now these guanines thus rest within the control sequence. And so we determined the sizes of the bands that are seen after the piperidine treatment so that we can determine the position in the DNA fragment of guanines whereby methylation prevented protein binding. Now, once we do that, we will, so this is so that we can determine sizes of bands consisting of restriction fragments cleaved by piperidine. Now, the next thing that we can do is then we can repeat this whole experimentation. So we can repeat experimentation, and we'll repeat the experimentation with chemicals that target adenine, thymine, and cytosine. So we target adenine, thymine, and cytosine. And that way we can deduce the precise position of the control sequence. And so this facilitates the deduction of the precise location of the control sequence. And this concludes this discussion on modification interference assays.